now officially in lab time. Um, so I am assuming that people had a chance to do those little tests for the general sensation. So I do want people to share some of their actual experiences around 2-point discrimination or touch localization and what you found and whether or not you found it surprising. Does anybody want to share their experience there? Or I can call it.
For the temperature one, I put my hand in the warm water,
Uh-huh.
and it took a long time for me to not notice it anymore. It's only like 50 seconds.
So, but after a couple of minutes, you probably didn't feel the warm water much.
Yeah, then I didn't, and then it was really crazy putting my hands both in the same bowl. It was like one was hot, the other one was cold. It was interesting because I
Yeah.
remember once I did the Alcatraz swim without a wetsuit, and I remember when you afterwards when you take a shower, you'd hop in the shower and it would be cold and it would feel like hot, really hot. So it's kind of the same thing
I
going on.
did that swim too with no wetsuit.
Yep.
It was terrible.
I loved it.
Did everybody have a chance to do the temperature adaptation where you put both hands in the lukewarm water? Was it surprising?
Yeah, it was really weird feeling. Took both hands in the same temperature, but it was really strange.
Right. I mean, I like these to give you a real sense that, of what your perception is, isn't necessarily some true readout on what's really out there. It's kind of filtered by your nervous system and your receptors. And the same thing with putting the coins. How long did it take for coins to like not even be felt if they're sitting on your arm?
It felt like a really long time to do the coins. It took me
Yeah,
like
it felt really long.
Right, what does that mean, really long?
It took about like 3 to 4 minutes for me to not feel any more coin on my forearm.
Well, that's interesting. I mean, did you make sure they were insulated? Because a lot of times what you're feeling is the coldness.
Yeah, I wrapped a tape around the. Well, first I tried with the quarter, and I thought that was a little heavy, so I thought it might take a little longer. So I tried with the penny, and I wrapped it in a tape, and then tried it that way. So it took me quite a bit of time, actually, like 3-4 minutes.
Okay, typically it's quicker than that. Typically within like about 15 seconds or something, people don't feel anything. How about what are other people's experiences in that?
The coin for me was about 20 seconds.
Yeah, it's, and again, it's different people for sure.
Yeah, I looked away, and so I tried everything what the procedure said, to eliminate any kind of sources of variability. So,
What about 2-point discrimination? What I can just like, I'll do like the random, like, I don't even know who I'm on. I am on Rosa Lomelli. Rosa, what did you get for your face in terms of 2-point discrimination? The closest that you could feel as 2 rather than 1.
I did it on my daughter.
So what did she get?
So, she was more sensitive in her palm.
But so
in the fingers.
What value did you get for the face?
Okay, for the face, I got to the point where she can feel 2 points. It was 1.5 centimeters.
Yeah, so that's typical.
Mm-hmm.
So the thing is, that's about 1/2 inch.
Yep.
This is something that most people really, if you ask the random like person on the street, how sensitive is your face? You think, oh, my face is pretty sensitive. If 2 things were like a 1/2 inch apart, I'd know for sure it's 2 and not 1. But they have to get almost like that's a little over 1/2 inch apart before you actually can tell it's like 2 instead of 1 on your face. Like I know like the face particular is something that is often surprising. You know, where did people find the finest discrimination?
My palm and fingertips for me,
Your fingertips,
Yeah.
and my palm.
Yeah, hands.
fingertips tend to be the best. You know, so if we go, you know, if I'm going to now go and share the modules. If we go into kind of class data, which you should, you know, use your data in your discussion, but then always you compare what you found to the general trends across many, because one particular person could be idiosyncratic. Could either be that person responds differently than most people, or there could be something in your technique that introduced some source of variability you didn't even realize. But you should have the group data as well. So if we go into general sensation, what? Where to go? Where to go? Oh, come on, little puppy dog. There it is. Doing the general sensation lab. If you go into general sensation spreadsheet here, you can open this up and oh, where is it? Come on. There it is. Okay. I gotta stop sharing that and do a new share. So here is this data, and we can like zoom in a bit. You know, here you can see across many different groups, 2-point discrimination. This also has tactile localization numbers. It also has adaptation, the time to not feel something. And then at the bottom, I have the, let's see. You know, 2-point discrimination. If you take a look, again, fingertips and lips in general, you know, you can tell within a couple of millimeters if it's 2 or 1. But when you get to the face, it's bad. Again, 1.5 millimeters, that's over half an inch before you can really tell. Back of the calf, these things can be over an inch apart and you don't know if it's 2 or 1. You know, so kind of just really driving home this point that unless you have the receptors to convert the stimulus into a message for your nervous system, you don't even know it's there. What do we call that process of converting the stimulus into an electrical signal?
Sensory transduction.
Sensory transduction. You've got the receptors in your skin converting that touch, that physical stretch of your skin into some electrical signal. Tactile localization, since it's based on the same density of receptors, is going to be similar. Did this surprise anybody when you kind of used a little pen to mark where someone had been touching you? Was it, did it feel like, oh yeah, that makes sense? Or did it feel like, whoa, that's weird?
It was kind of weird. Did not, well, couldn't pinpoint the first pin marker.
Yeah, I mean, I know for me, like the first time I did this and I like, I'm pretty sure that's where I was touched. And then I look, it's like, holy crap. I'm off like by a 1/4 inch here. And it really felt like that was it. You know, so just realizing you have this kind of limitation in how well you can detect things and localize things and detect 2 versus 1 and all that. And we'll see that you get similar things going on in your other senses. Adaptation happens not just in temperature and not just in touch like we saw. Your visual system adapts, and adapts in some really trippy ways. I'll just do this now because I think it'll probably work over the, I'll link to the YouTube thing later so you can double check it. But if you've never done this, this is really kind of cool. Like if your visual system is getting used to things looking a certain way, moving a certain way, you adapt to that. And then if you look away from it, your visual system is now going to overcorrect the other way. Just kind of the same thing with the temperature. If your hand's been in the warm water, now the lukewarm water feels cool. If your hand's been in the cold water, you've been swimming into, Alcatraz, you get in the cold shower, it feels warm. If your eyes have been seeing things spiraling around and that becomes the new normal, and then you look at things, then the thing that should look like it's not moving might look different. So I'm going to, for this thing, you look at the screen. I'm going to share this right now. Where is it? New share. So for this thing here, you can see the middle where it has that little 40. That's just a timer. So you're supposed to watch this for 40 seconds once I get it started. And you don't look away. Just stare right at the middle where the numbers are going down. And hopefully this is big. If you can, as much as possible, this should be like filling your screen. This should hopefully fill your screen. And I'm going to run it for 40 seconds. And when you're done, you can look at the screen. I recommend look at the back of your hand. That's, for me, that's one of the weirdest things ever is watch this for 40 seconds. And then as soon as it's done, just look at the back of your own hand and see what you see. So we're going to start this thing. And again, just stare straight at the middle. And it's got cool music. And it might be jittery. It might be too jittery. I think it might be too jittery. Do people see anything? Or if not, I'm going to link to it and I will have you do this at home and then write something down because it's really, really, really weird and cool. I think it was too, it wasn't smooth. I was looking at it on my iPad streaming and it didn't have the nice smooth spiral. So we'll do this. We'll do this on your own where you can see it better.
No, I see lights.
That's not what it is.
White.
That's not it.
The back of my hand wasn't moving, but when I kept looking at the clouds afterwards, the clouds were moving.
So we'll do this at home. I'll set something up for you guys to do. So it'll be cool. All right. So other things about the general sensation lab. Just make sure in your lab write-up, you kind of describe your personal observations. In fact, let me write this down. Now that I've been grading more and more of these labs, I'm finding more and more of this split between people who seem to understand what's going on and some people who are really lost. So when submitting PDF of your write-up. So one thing, when you're submitting a PDF of your write-up, like the ones that are for the pre-labs, that's just the pre-lab. That is obviously the objective, hypothesis, and sources of variability. And those, you got to get those before you can do the main lab. There were a few train wrecks where people never finished the pre-lab properly on labs, and then just wrote up the whole lab, which ended up being a total mess because they didn't actually even know what the whole lab was about, because they never actually solidified what are the independent and dependent variables here, what is our objective. So don't do the full, it doesn't matter so much for general sensation because that one doesn't have a pre-lab. But in general, as we go on, don't write up your whole lab if you didn't actually get your pre-lab approved. That point of doing, even though you get ultimately 0 points for the pre-lab, that's what you need as the springboard to actually do your lab properly. And in fact, I start taking points off if you just can't get it, because I need to have people pay attention that you got to do this. This should be complete. When it says do the complete lab, it has to include your objective, your hypothesis, the methods, has to have your data, your analysis, if any kind of graphs and things, and then your discussion. The data and the analysis are usually figures and tables. They have numbers. You need to have numbers, like as we see in table 1 or figure 3 or whatever, and your discussion needs to integrate those numbers. You can't just say like, yeah, we found this. It's like, what did we find? Show me. Lead me so I can see the graph you're talking about. And use quantitative data. Instead of just saying, yep, this thing moved faster, you can say, as we see in table 2, the potassium permanganate, had an average rate of 4, whereas the methylene blue had an average rate of only 2 millimeters per, hour, and therefore we saw that it's going like twice as fast. If you have data, like when you're talking about the skin, 2-point discrimination, talk about actual numbers. Like, don't just say, oh, the lips were more sensitive, done. It's like the lips were more sensitive, as we see, they could detect down to 2 millimeters, whereas on the face, it was, you still only felt like 1, even if they were, 7 millimeters apart or whatever. So use, be concrete, and then refer, like, as we see in figure 3, the lips were much more sensitive, having 2-point discrimination that was down to the millimeter level, as opposed to the calf where they couldn't feel them even when they were 20 millimeters apart. So stuff like that. Make sure your discussion assuming if you have hypotheses, make sure you are referring to your hypotheses. Make sure, your objective has your independent and dependent variable, particularly in some of the characters who never quite figured out the pre-lab. They wrote their whole discussion and never actually even addressed, the dependent variable. So it's like, here's all my discussion of what all this experiment meant. And they didn't even get at the core question that we're talking about. So make sure your discussion is really looking at what is that relationship between the dependent and independent variable. What does our data say about it in a very explicit way? How does it tie into the bigger picture? What are sources of variability? Things like that. So just putting that out there. Some people's lab write-ups were like just amazing. Some of them were you know, were really not. And some people just didn't. If someone submitted something where it's just not complete, I requested, please scan the whole thing and I'm giving you the benefit of the doubt that you just didn't understand. You needed to submit the whole write-up there. So please make sure you submit it so I can give you a proper grade on it. You know, that being said though, for some of the people who find like literally like I keep sending little things like, you know, this was due a week ago, this was due 2 weeks ago, and people just don't respond. At some point, I have to give you a 0 because, you know, we got to move on. So
this is a question to everybody. Is anybody experiencing any uploading issues on the Adobe scanner? Like when you upload like a full complete lab, only the part of it gets uploaded, and then you find out later that that's the case.
I haven't noticed that.
I haven't, Jamba, either. Neither. No.
most of them have been coming through pretty well. There's been a few cases where the settings I think are off. Sometimes about the color balance where everything's really like washed out and I can't really even make out what things are, or where the sometimes either the resolution's too small or too wide. Sometimes people, one particular case, someone turns in things that don't even fit on my screen. Another case, somebody turns in things that are so small, even at the maximum zoom, I can't even like barely read what they're trying to do. So in there, I'm not sure. There's got to be some settings. I've sent some feedback to people about trying to work on your scanning settings. But just kind of, you know, we've still got a lot to go in this class in terms of these write-ups. Even before, you know, in the today and tomorrow, no, today and Thursday, we're going to be doing the vision and the hearing and the gustation and olfaction. Those are going to have write-ups that you're going to have to turn in. The second half of the semester is going to have quite a few major write-ups. And those write-ups are worth a reasonable part of your grade. Plus, those write-ups are going to give you what you need to do well on the lab exams. You know, you're going to have your first lab exam coming up. Where is it? On Thursday. So a week and a half from today is your first lab exam. And in general, if you've been doing your labs well and doing these write-ups well, where you're very clear about what is the objective, you know, what do the data have to say about that, you know, and you've written it clearly in your lab notebook. How did we do it? What did the test tubes look like and why did I interpret it that way? It's an open notebook exam. So if you've done a good job on your notebook, then the lab exam is pretty easy. It's all there for you. Whereas if your lab notebook is a train wreck, it's not going to help you because whatever was scattered and, you know, not understanding the lab when you were trying to write it is still going to not make sense when you're taking the lab exam. So you want to do this because it's worth points. You want to do it because it's going to help you do well on the lab exam. And you want to do it because, you know, one of the skills that we're working on is how do you kind of more rigorously, you know, describe things, collect data and interpret what it means, draw responsible conclusions, you know, which, like I said, is a useful skill if you're doing science, but it's also a useful skill if you're just out in the world trying to figure out like what works, what doesn't work, and, you know, trying to be the best person you can be in a clinical setting or anything, like trying to make sure you're solid in your observations and your conclusions that you draw from your observations. So anyway, blah, 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 blah. Any comments, questions about the bigger picture of lab notebooks before we move on?
You said that lab exam's next Tuesday?
Yeah, it's on the schedule and it's going to be next Thursday. Right now, my plan, I think I'm going to be able to stick to it, is to do the vision today. And then on Thursday, we'll do hearing and the chemoreceptions. And then next week on Tuesday, we'll tie off any loose ends we need to tie off in case we don't get through everything. We can discuss the different things and have kind of a review of everything and what the expectations are on the lab exam. And then on Thursday for lab, rather than doing a lab, it's going to be another one of these Proctorio-based tests, but it will be about lab material. You know, it could include pictures. Obviously, normally it would include actual test tubes that you'd have to look at. Instead, it's going to be like pictures of stuff you might have seen in lab, or, you know, questions about specific labs and data and what does it mean, or maybe your particular lab, what did you find and does it match what you expected, or whatever. You know, it goes into the specific labs that we've done, as well as kind of the basic processes that we've been talking about in the lab. And I'll mention one last thing. You know, lecture, lab. There's kind of these two parts of this class. This kind of lecture part where we're talking about more the theoretical and esoteric nature of things. Lab, where you're actually doing more of this practical hands-on, and also these kind of skills about observation and interpretation of data and analysis. You know, both of them are important. They do like intertwine, but they're also something fundamentally different about them. And you need to pass both of these. You can't get like, oh, I got an A on all my lecture exams, but I never turned in a lab, but it averages out to a C. And it's like, nope, you're going to fail the class. You need to have passing scores on both the lecture and the lab materials in the class. You need to be passing the lecture material as evidenced by the lecture exams. You need to be passing the lab material as evidenced by your lab notebooks and your lab exams. So just kind of putting that out there to get a final passing grade in the class, both, you know, because I'm thinking some people are thinking I'm gonna like blow off all the labs, but I'll still do okay on the lecture stuff, so I'll still pass the class. But that's not it. You need to pay attention to the labs. The labs are doing something fundamentally different than the lecture stuff that is important, that are part of, you know, the student learning outcomes, what you need to know to move on to the next level. Okay, just kind of putting that out there, just because, again, for a lot of people, there's this great, people are just rocking it. But some people are, and some people are completely not rocking it. But then some people are in this weird zone where they're kind of quasi, and one side looks fine, but the other side really doesn't. And you really need to pay attention to both. Okay.
I had a quick question about that lab exam. What is it going to go up to? Which labs is it going to include?
It will go through this week, through the sensory labs, the vision that we're doing now, the gustation and hearing, which we're going to do on Thursday.
It's going up through general sensation, and that's the last one, or vision as well?
No, all of these ones.
All of them. So
vision and hearing and olfaction and gustation. So everything through this week.
Okay.
Professor, and we're going to use Proctorius, but it's open book, so we're going to be able to see our.
Yeah, so open lab notebook. You
Exactly.
can other things, but you can use your lab notebook.
Ah, okay, perfect. And we scan it and then good.
What's that?
We will scan the lab notebook for proctorials.
Yeah,
And
it's
okay.
fine. When you're showing where your room, you can just have your lab notebook there. That is
Okay.
that's
I have a thing, I know it's weird, but the lab book that I bought, I don't know why it came with 92 pages, so I'm almost done, but I have a new one that I'm gonna start using. So do you want me to staple it to this one or to separate?
write volume 2 on it or something.
Huh? Okay, okay,
Yeah.
perfect, thank you. Because I will use it probably to finish a special census.
I see. Okay.
Okay, thank you, Professor.
Yeah, no, make sure you don't get ones with like the carbon copies and stuff, because those are the ones that, you know, if you normally you have 150 pages because you have, you know, the front and back and everything.
I don't know why this is hardcover. I bought it in a rush because I, whatever, and it's, but the other one I bought is the regular that I see with the green pages.
Okay.
So just so I'm clear, so for before our lab exam, we're going to need to complete the write-ups for vision, hearing, olfaction, and gustation.
Absolutely.
Okay.
Yeah. So, you know, the write-ups for these ones, though, hopefully the other ones you've been keeping up with. They're either done or almost done. And you're going to have all weekend, you know, to finish writing up the ones we're doing this week. So, and again, these are not ones that have heavy hypotheses and objectives. These are more doing things that demonstrate different concepts. So vision is what we're doing now. So similar to general senses, you know, we haven't gone over vision in lecture yet. So we need to talk about some of the background before this is gonna make sense. So obviously what is the stimulus for vision?
Light.
Light. So light, and we have, you know, the organ that is going to do this is your eyeball. You know, I'm drawing here kind of an eyeball from the side. If this was like, if you were looking at an eyeball from the front, you would see, you know, like a person with green eyes. So if this was an eyeball from the front and then I took a slice through it, I would have this kind of clear window here, which is where the light's coming in. That's the cornea. If you remember from anatomy, it's part of the fibrous tunic. The cornea is just the clear window that enters into the eye. Then we had the vascular tunic, if you all remember that. Again, we're going to talk about this more, but for right now, main thing you need to know, there's the iris. Right, if I look at the iris in cross section, this is the pupil here. The pupil is just the hole where the light is coming in here. So the iris on the back is going to be the retina. This is going to be the rods and cones back. These are my photoreceptors. We're going to talk about them in more detail later. Again, what does a photoreceptor do?
Receptor to light.
And if you're going to give me more detail, what does it do?
Converts the stimulus of light into an electrical signal for your brain to process.
Exactly. They're going to convert light into electrical signals, which then go out your optic nerve. I'll talk more about the wiring later, but the basic idea they are all going to send their signals to the brain via the optic nerve, cranial nerve 2, if you remember. And there's kind of a weirdness in that all the axons actually are on the surface, and where they burrow out the back of the eye to go to the brain, they create this place where there are no photoreceptors, there are no rods and cones. That is going to be important. That place right there, where there are no rods and cones because where the optic nerve comes out, that is going to be called my blind spot or my optic disc. So that's kind of important to realize, that images come in, if they're landing on the retina with the rods and cones, you know there's something there, you can see it. But if that image happens to land on that one particular part of the retina, where it's the blind spot, where there are no rods and cones, you actually don't have any information about the image. And we're gonna look at that. It's gonna be the first thing we look at in terms of our lab. Like, well, what happens? I mean, obviously you look around and you don't always think of like, oh yeah, of course, I know there's this big spot in my vision where I don't see anything. You know, your brain makes stuff up. So when you look around, it just all looks like you see lots of stuff. But in reality, it's not true. The other thing is we have the retina, the thing that does the phototransduction, the sensory transduction. But the other thing we need is we don't want to just know whether light or no light. We want to know kind of the details of the image. So that means we have to bring it into focus. So the eye is like a camera. And it's very much like a camera, that you have a big lens. Just like your camera has a lens that is going to bring your image into focus. You know, in the retina, this is kind of more like the film. You know, or, you know, in a digital camera, it's like the CCD array on the back, whatever. But so we have two things going on. One thing we're going to be looking at today is thinking about kind of the sensory transduction part, the rods and the cones. And that's just about whether you see or not see. We're going to talk about how the cones detect different colors and things. The other thing we're going to worry about is the lens. How do we bring the image into focus or not? Can we see it clearly or is it blurry? The lens is operated by these little ciliary muscles, and you've got these, they're called suspensory ligaments that attach the lens to these little muscles here. And the basic idea here is using these muscles, you can change the curvature of the lens. Again, I'm not going to get into the details right now, but when the muscles are relaxed, the lens tends to be more flat. When the muscles start tensing, the lens can get more and more kind of curved. So I need to talk actually a little bit about that aspect of the lens. So the lens is basically we have our image, we have our light coming in. The lens is going to bend the light rays so they land right on the retina and they're in focus. So again, we're going to talk about this. We call this converging the light rays, when the light rays bend inward. So the image comes into focus here right on the retina. When you have a lens that is kind of bulged like this and converges, we call this a convex lens. So convex, and you can have convex, you can have more convex, more bulgy or less bulgy. And the more bulgy it is, the more it bends the light. So if I have my light rays coming in to a convex lens, they'll bend in. If I have the light rays coming into a more convex, they'll bend tighter. They'll come in, they'll bend. So we can bend the rays. We call this converging. We can converge the light rays more or less depending on how curved the lens is. And you have smooth muscles, the intrinsic muscles of the eye. You can actually adjust the curvature of the lens using these little autonomically controlled muscles within your eyeball. So you have the ability to change the curvature of your lens to bring things into focus or out of focus, which is going to be important in a few moments. The other kind of lens that we should mention, because it's important for corrective lenses, is the idea of a concave lens. I won't, there it goes. So here's my concave lens. And when the light comes into this, it diverges. Again, this is not something that is naturally, this is not a lens inside your eye, but this is going to be the kind of lens that you use as a corrective lens. So if you're, we're going to see specifically, like if you have myopia, you're going to need something like this, and we'll talk about why. So you should understand the basic kind of lenses and what they do. I mean, it's kind of silly. The way I remember when I was first kind of learning this, concave lens, like if you were a bear trying to find a place to hibernate for the winter, you know, where are you going to go? You're going to go find your little cave here. You're going to, you know, there's no place to go. If convex lens, there's no place to walk into to go hibernate. Concave lens has a little place to, a little cave in there. Anyway, so lenses, and back to our eyeball. So when we start talking about visual acuity, we're going to be talking about more detail, the details of how the lens either converges more or less the light coming in and brings it into focus to allow you to focus on things near, focus on things far. I'm going to be looking at that, and we will all be looking at that in more detail, but we're going to start by thinking more about the retina. The first few parts of today's lab are looking at things that are going to help you understand the retina a little better. And the very first part, in fact, let me, demonstration of the blind spot. So if we go share screen, going to, hold on. Screen 2 here. You know, Vision Lab, here's just the pages of the Vision Lab. Again, as I was just mentioning, the blind spot is this area on the back of the retina where you don't have any photoreceptors. So optic disc or blind spot, there's part of the back of your eye with no photoreceptors. So if the image lands on that blind spot, you don't have any information about it. So this part here, this is in your lab notebook, hopefully, or lab manual. Tell me, does anybody not have a picture, not have a copy, a printout of their lab manual in front of them right now? So everybody get out this is page 107 of your lab manual. So everybody got that? Let me see y'all. It's like a lot of people are just black boxes. It would be nice to actually see people while we're doing this. Yeah, because it's more fun to actually like feel like we're in a class. So the point of this, if you've never done it, it's really weird. Even if you have done it, it's every time I do it, it's still like, whoa. So the idea here is you hold the page, somewhere about a foot away from you. And the basic idea is you got to get the angles just right. And as you move the page closer or farther away from you, the different letters will land on different parts of your retina. So you close your left eye. So the left eye. And then, so using your right eye, you look at the little R. And then you move the page closer or farther away, just looking at the R, but noticing the L. Try to, and go slow. I see Taylor going really fast. Because the sweet spot, you can go, it's going to be closer than that, Taylor, probably. You want to come a little closer in.
I don't know. I don't see the L right here.
Wait.
Yeah, when I get the page closer to the eye, the L disappears.
Right, that's it. So do you see when the L disappears?
Yes, perfect.
But if you push it closer or farther, it should come back and it should come back. Right?
Yes,
So
super clear.
So is there anybody who does not see that?
Very good.
Like Rhea, can you see it? I can't hear you.
No, I don't have, like, I already have vision problems and I don't have my glasses with me and have to wait for them to come back from the optometrist. I had to get new ones.
You can't. Wait, no, this is
Like when, I know, but like when I get the paper close enough, like it just is all a blur.
Okay. So how about Kelsey? Did you see it? Yeah, so like in your write-up, you are going to want to measure what is that distance. So, and you might get some help. Once you're there, somebody can take a ruler, and so you're going to write what distance is that. And then again, you can do it on the other eye, but you have to do it the other way. And again, it's kind of a trippy thing. It's basically the brain does not know what's there, but it fills it in. Like it's white above, it's white below, it's white to the left, it's white to the right. What are the chances it's not just more white page there? So your brain fills it in. We're not going to do it right now because I want to keep us rolling along, but you are also on your own. Do the next couple, because you can actually fill even more complicated things. You can do these ones, you fill in lines, you complete the converging spokes. You know, it's really kind of a trippy thing. I included a couple of videos that you can look at that also run you through. One of them does a blind spot thing where you get to see that it actually fills in colors in the background. It doesn't just fill in white backgrounds, it'll fill in colored backgrounds for you. Your brain is trying its best to make sure that everything looks fine, even if in fact there's a hole. So that's going to be the first part of the lab, is looking at the blind spot in more detail. Again, for this lab, kind of like the general sensation lab, I'm going to kind of lead you through things, but once we're done, there's going to be stuff you're going to have to do on your own, because a lot of this is stuff where you do and you measure each other, and we can't do that. Like, you know, so instead, I'll have you do it in the comfort of your very own home. So if we get back here, demonstrating after images. So for this, we need to, let me talk a little bit more about the photoreceptors, the rods and cones. Again, we're going to look at these in more detail as we go through the semester. I am just introducing them for the first part. We're going to talk about them in much more detail, look in excruciating detail how they actually convert light into electrical signals, look at the different kinds of wavelengths that they respond to. But for right now, they're photoreceptors. There are rods that are monochromatic, meaning they only respond to the absence or presence of light. They don't give you details about the actual color of things. They just tell you whether or not there's something there or not. Like at night, these are what are in effect. They work well in dim light. So that's why in dim light, things don't look very colorful. It all looks washed out. And we're going to talk about rods in much more detail later as well. The cones, which function in bright light, that are kind of in the center of your eyeball or center of your retina. These, there are 3 different versions of them. You have the red, the green, and the blue that respond optimally to light of these different wavelengths. So if you have blue light coming in, the blue cones are the ones that respond, and therefore that's how you know it's blue. If you have red light coming in, the red cones are going to respond, and that's how you know it's red. Same with green. And then everything in between is just a mix of these. It's a little different. Like in school, you were always like, you had the primary colors were like yellow, blue, and what was it? Yellow, blue, and red. So that is mixing pigment, right? And if you wanted green, you got yellow and blue, blah, blah, blah. When you're mixing luminance, when you're mixing light, it's different. So it turns out when you mix all of these together, you get white. Right? If you mix paint, if you mix red, yellow, and blue paint together, you get like mud. But with light, if you're mixing luminance, you mix all these together, you get white. You mix other combinations of these lights, you get all the other colors. So these are the 3 colors that you need to detect every other color. If you are lacking one of these, you are not going to be able to discern something that differs only in that particular type of color. So that's what we call color blindness or color deficiency. You can still tell if is that a dog or not or whatever, but is it a tawny dog or a grayer dog? I don't know, because I don't have the ability to tell things that are different that only differ by shades of some component of red, right? So color blindness is when you have a genetic issue where one or another of your cones is not there. Color blindness is we call a sex-linked trait. What does sex-linked mean when we're in this context?
Male and female.
That it's genetically linked. You can have it from your mom or your dad, like from X or Y.
So no, so these sex-linked, this is on the X chromosome.
I just X.
So, and things that are on the X chromosome are particularly interesting because if you have a mutation, for instance, and you have a broken copy of the gene that makes the red cone on an X chromosome, if you're a guy, you're done, right? A guy is XY, right? So if you have a bum version of the gene to make the red cone on the X chromosome, your other sex chromosome's a Y, so you're colorblind. Whereas if you're a woman, you've got two X chromosomes, right? Women are XX. So if you have a bum version of the red cone on one of your X chromosomes, the chances are you got a good copy on your other one. So color blindness is very rare in women, right? For a woman, you have to have two bad hits of the gene on both of your X's. Whereas for a guy, you just need one bad version and you're colorblind. Like in teaching this class for 25 years, it's not uncommon to have colorblind dudes in the class. I have yet to meet a colorblind woman. So colorblindness, sex-linked because it's on the X chromosome. And I'll show you, we'll go through the tests to test for colorblindness. You should all do it. Although, like I said, if someone's going to actually find they're colorblind, it's going to probably be a guy and not a woman. The other thing about these rods and cones is, they take in light. They're called rods and cones because they actually kind of look kind of like rods and cones. And if you take light comes in and then the electrical, it's a stinky picture. Let me try this again. So light is coming in and then they're releasing all their glutamate, their neurotransmitter to say, I've seen light, or actually we'll see it's actually the opposite, but whatever. The light comes in and they convert light into these electrical signals. You can overexcite them. If you shine a bright light on them and then you get rid of it, they'll keep firing off for a moment. That's like you stare into the sun and you look away and you still see a dot, right? It's because those receptors are still like, even though the light isn't there anymore. So you can have after images that happen if you kind of excite them. And then even if you take that away, if it's been this really bright excitation, the response can continue on for a little bit. So you still see dots, even though there's nothing there to see. But then you get this really trippy thing of what's called the negative after image. So if you really excite this thing a bunch, and it's going and going, you can kind of deplete it. And then when you finally stop exciting it, it takes it a while before it's active again. Then let's excite it again. It's like, I need some time to recharge. And then finally it starts coming back. So you have this thing called a negative after image, where if you've been overexciting the photoreceptors, they actually have this period where they don't respond. And I'll show you, there's actually a really trippy little illusion that I'll show you in a few moments. For instance, if you overexcite the red, let's say we're putting a lot of red light in and all your red cones are getting really excited, but then they get kind of depleted. And now I take that red light away. The red cones are actually kind of depressed. They're not responding as much they normally would. So everything shifts and looks more green than it would have otherwise. Because now, normally the red and the green would be balancing each other out. But since the red is no longer as active because it's been overstimulated, now all of a sudden the green looks emphasized, even though it's not, there's no emphasis in the red in reality. I mean, in the green in reality. Let me just show it to you. Let's just do it. I'm gonna go share screen again.
Is this only with cones or is this with rods and cones?
This is gonna be with the cones because the rods don't have different colors. And also the rods, we're going to talk about, the rods basically kind of get bleached. They're not designed to work in, they take a much longer time to get ready to go again. The cones are more optimized to work in bright lights, and they get like, you get a bit of a negative after image, but then they're good to go again in the bright light. All right, so we are going to go into doing the vision lab. So here, demonstrating after images. This is so I think this should work for you all. So the basic idea here is, so I want you to see, if we go further down, the image of the bee that you're going to see afterwards is a black and white image of the bee. There is no color in this image of the bee. The only color that you're ever going to see is a negative image of the color. Like it's like blues for red, blues for yellows and reds for greens and stuff. So as you're staring at this negative image of the bee, you are going to be adapting or you're going to be getting this, all your rods, oh, you got facts, I'm sorry. Whoop. All of your cones are going to be getting excited by these different colors, and they're starting to get depleted. So by the time we finally stop looking at this negative picture of the bee, your eyes are going to have their color balance off. And this is designed in such a way that it's going to mess up your color vision in that the black and white picture is going to look. So stare at the middle of this. Don't move your eye. You really want it to be resting on the same place on your retina. Keep staring at it and notice what the bee looks like as soon as the color stops. This should work because we don't care about the motion as much. Did you see for a moment that the bee looked like color? That was a negative after image. And again, you can just do this at home. It's this weird trippy thing of the cones, all of us, for a few moments stopped responding. You could do it just if you shine a bright light and you shut your eyes, you're going to see kind of like a black spot where the bright light used to be because those photoreceptors are stopping responding. So, in your lab write-up, you can do the thing with the flashlight and describe what you see. Here I said also do this cool YouTube example and record your observation in your lab notebook. So I just want you to get this idea of what the after image is, what a negative after image is. Testing for color blindness. This is these special plates. Okay, this guy is kind of hilarious. For some reason, I keep thinking this is like Steven's like long lost cousin or something. He looks.
So are you colorblind? Well, in
Doesn't
this
it-
video, I'm going to be walking you through the Ishihara colorblind test so that you can figure out if you are colorblind or not. Let's take a look.
So I'm not gonna, so basically you should watch this, and he goes through each of the pages here.
Color vision, they should see an 8 on this first page. However, somebody with color.
So he's gonna go through, these are the pages. Normally we would have these for you in the lab, but this actually works pretty well. So you're just gonna watch, and you're gonna look at each of these plates. There are 14 in all. So in your lab notebook, you should put a space for plates 1 through 14, and you should say what number you see and whether that means you have normal vision or you're colorblind. People who are not colorblind, you know, when they look at this, they see an 8. And most of you probably see an 8 here. People who have certain forms of color blindness, these orangey dots just don't look any different from the green dots, and they just see, oh, it looks like a 3. They don't see an 8. You know, but other people see an 8. Same thing with this 5. You know, some people see a 5, some people see, you know, let me clear.
Color vision deficiency, like a red-green deficiency, should see something closer to a number 3. And then on the other adjacent page, you should be seeing a 5 if you have normal color vision. And then again, somebody with some sort of red-green deficiency will see something closer to a number 2 hidden in there. Now, on the next page.
So this gives you a sense. I mean, and it's trippy. And what is kind of fun, actually, let me get, you know, because you always think of like, you know, color blindness as a deficit, but like this one here, this one here, if you are not colorblind, all you see are a bunch of random dots. But if you are colorblind, a lot of these patterns disappear and you see like this clear number 2 in the middle of it. So just because somebody is, you know, colorblind, you know, actually in some ways, some things will stand out in the world that don't stand out for normal or for normal vision. So in your lab notebook, watch this video, write down your results. There are 14 of these plates, you know, and draw a conclusion about whether or not you're colorblind or not. If you are sheltering in place with a guy and you are not, if you're a female and there's a guy around that is willing to do this, it could be fun checking them out because there's more likely to find some color blindness there. Because again, women in general don't have it because, you know, you got 2 X chromosomes, so it's really unlikely you've got 2 bad versions of the gene for the cones. Any questions about color blindness?
So the 2 X chromosomes on a female, if one of the lead to colorblind, or both X chromosomes have to be bad?
In a woman, right? Normally you have 2 alleles, right? Your genome is gonna, your genotype, you have 2 alleles of every gene, the one from your paternal and one from your maternal version of your chromosomes. So most of the time you have 2 versions of everything, but except for the sex chromosomes if you're a dude, because you only have 1 X chromosome, so you only have 1 copy of all of those genes. So if you have something that's gone wrong on one of those genes, that's it. Obviously on the Y chromosome, there's nothing essential to be a human because women don't even have Y chromosomes. The only thing on Y chromosomes are things involved with maleness. So So that's part of the lab, testing for color blindness. So again, these are things you can do at home. And these are things that are involved with trying to understand more about the retina and the photoreceptors and, you know, the other parts of the lab that we're going to do now are going to be dealing more with kind of the image formation and focusing, trying to make sure that you have a clear image on the retina. You know, the retina can tell if there's light or dark, or like light or non-light, but you want to have it in focus. So visual acuity. Visual acuity is the idea of how much detail can you make out with your sense of vision. And that's going to depend on using the lens to bring the image into focus on the retina. So I am going to look at this in more detail now with you. We've got, yes, 12. So I'm going to spend a little bit of time talking about kind of the theory around normal vision versus nearsighted, versus farsighted. Then we'll talk about kind of the practical clinical tools people use to assess that. All right. My glasses now. So eyeball, kind of retina. I mean, not the retina, that's the cornea. Again, kind of the retina is on the back. And then the lens. And the light is coming in. So there is a kind of assumption. So I'm going to be a little fast and loose with my optics here. But the basic idea, if you have an object that's far away, the convention is 20 feet or farther away. Let's say you're looking at, I don't know, let's say you're looking at I usually use a deer. Let's put a deer there. There's a deer standing 20 feet away. The basic assumption is that the light rays are coming in parallel from things that are far away. That's the basic approximation. I want them to converge to come into focus on the retina. When your eye muscles are at rest, the lens has just the perfect curvature to make these land on the retina and come into focus. So this is, you know, kind of lens at rest. It has just the right amount of curvature to make things that are far away come into focus. And what we're describing here is called the emmetropic eye. Emmetropic is normal vision. So the first thing to kind of make this all make sense, you have to remember we have a lens which converges light rays. The light rays are coming in, they're getting bent by the lens so that they land just in focus on the retina where you actually convert the image into a nervous system signal. When things are getting closer, then you no longer have this assumption of the lights coming in parallel. So let's say, in fact, people are talking about the video of that cougar. It's like, oh my God, that cougar is getting close. The light rays from something that are close are actually kind of diverging. They're coming out. So this is close. Which means if you don't change the curvature of the lens, it doesn't bend them enough to make it focus on the retina. So the image on the retina is going to be all blurry. So how could I make this mountain lion come into focus on my retina using my lens?
This muscles of the lens contract or dilate, and they put curvature.
Exactly. I can contract those muscles, increase the curvature of the lens, and now that is going to bend the light more and put it into focus on the retina. So we call that accommodation. So the closer something gets, the more the lens has to accommodate in order to keep it into focus. So does that make sense? Because this is kind of at the core of everything else. If this makes sense, then the rest of it will make sense as well. Obviously, accommodation can only keep occurring up to some point. At some point, the lens has some maximum amount of curve it can do. So if you bring something really, really close, the light rays are so divergent as they enter, no matter how much your lens tries, it can't bring it into focus. So if you bring something closer and closer and closer, at some point, you can't, no matter how hard you try, you cannot bring it into focus. And that's called the near point of accommodation. This is closest, closest can be and still in focus. So does that make sense? It's like if something is 20 feet away, the lens doesn't have to bend at all. You can just be relaxed. As things get closer, the lens has to bend more and more to keep the image focused on the retina. But at some point, at this near point of accommodation, you hit the point where you have the maximum curvature possible, and no matter how hard you try, it's going to stay blurry. We are going to do this. You all are going to do this personally. You basically just have to get something that, something that you can actually look at. Like here I'm looking at like a pencil. It says 2H there, and I can bring it closer and closer. And at what point is it blurry or not blurry? Find that distance. How close can it be where I can no longer get it into focus? And then having your buddies, measure it using a ruler to see how far from your eye is that thing. What happens as you get older?
That lens starts getting bent with age. No, flatten.
It actually is. It's the opposite. As you get older, lens is less flexible. So you all see me wearing these little cheaters here. Like until I turned around 55 or something, I never had to wear any kind of glasses whatsoever. And now if I go to a restaurant, I better put these in my pocket or I'm in trouble if I'm trying to figure out what's on the menu. You know, why is that?
Because you're getting older and the lens, it's not as flexible as before. It's more rigid, so it cannot be bent by the muscles.
Exactly. So now if I see this deer far away, my eyes are just as good as they always were. But as things get closer and closer and I'm relying on my lens curving to keep it in focus as it gets closer and closer, you know, it used to be I could focus on something like a few inches from my face. Now, without these things, you know, it's about here is where it's like really in focus. You know, it's not even here. You know, you probably have seen this as people get older and they're like, well, what is this thing here? You know, so there's actually a word for it. The word for your near point getting further and further away as you get older because of this lens being less flexible is called presbyopia. Let me just write it on a new page. Opia. Which means old vision. So it's just as you get older, no matter what, your visual near point tends to start getting farther and farther away because your lens is less flexible and you have less of an ability to accommodate. You know, I will be sharing data for everybody so you can actually see what that looks like as you get older. So you, in your discussion, in your lab notebook, you should put your own data, but then also make sure you print out the data that I have included in the web page or on the module page. It says part 6, do at home, record your own data, but also discuss the group data in the spreadsheet that I've provided for you. When we do the hearing in the next lab, we're also going to see, you're going to pretty much from the moment you're like kind of late teens onward, your senses all start getting worse and worse and worse in measurable ways. When we do the hearing, we're going to be talking about presbycusis, like old hearing, as your ability to hear high frequencies starts just plummeting. So we'll be playing, or we'll be throughout the semester, we'll be seeing the senses becoming less accurate as you go on. All right, so this is just normal emmetropic eyes. Even if you had, again, your vision was perfect your whole life, you've still got that to look forward to, or you're probably experiencing it already if you're getting into your 40s and 50s and beyond. What about nearsighted and farsighted? Let's talk about those now. Those are when your eye just at rest isn't doing what we've been talking about. So let's start with myopia, which is nearsightedness. Basic idea is, here's your eyeball. Here's the lens. But in fact, let me draw this eyeball a little different. I'm going to draw the eyeball kind of more elongated. You know, and this is one way it can happen. So the eyeball is kind of elongated. The retina is back here. The lens is, like we've been talking about, the lens. And we're going to draw, here's my deer here at 20 feet. And the light rays coming in parallel, which is, again, the approximation for things that are 20 feet or more away. And the lens is going to bend just the same way that we saw last time. But because the eyeball is elongated, by the time the image hits the retina, it's actually going to be blurry. So you're looking at this deer and it's blurry, even though your lens is relaxed, and normally it would be in that kind of place where you'd see it in focus. Is there anything you can do to ever bring this deer into focus here?
Corrective lenses,
What's that?
like corrective lenses. So
So you'd need corrective lenses. There's nothing though that your lens can do, because your lens can only become more curved. It can't become less curved. And becoming more curved is actually going to make it even worse. It's going to bend the light even more. So this person here, without glasses, is never going to see this deer in focus. However, if you're looking at something that's closer, let's make it a little less stressful. Instead of being the cougar, let's just say we're looking at a flower. Oh, it's a beautiful daisy, right? And we talked about things that are close. The light rays start by diverging more as they enter the eye, which means that they are going to end up converging further back given the same lens. So it turns out that if you have something close to your face, you can still bring it into focus, because that pre-diverging nature of the image is going to allow it to end up in focus on the retina. So this person, this nearsighted person, myopic person, can bring something into focus if it's close, but they're never going to bring something into focus that's far away. And again, what kind of glasses, what kind of lens would I use for somebody like this to make them have normal vision?
divergent lenses.
They need to diverge, which would be concave. So a corrective lens for this person are going to be like this. So there's going to be this concave. So when the light comes in from the deer, it will have an initial divergence, more or less depending on how bad your myopia is. But they're going to have just the right curvature. So if your lens is relaxed and you're staring at something far away, it will be in focus. And now we're basically in the condition like the emmetropic eye. The eye is going to work like normal. Does that make sense? Yeah.
How surgery can correct myopia if the eyeball is like more
Right.
or
It's
elongated?
lasers to sculpt the cornea to create a lens.
Okay, got it.
So myopia, hyperopia, or farsightedness is kind of the opposite. So imagine the eye is kind of squished. And we're looking at the deer. Right. Light rays coming in parallel, converging like they would normally, but the eyeball is short. So again, this deer is not in focus when this person is looking at it. Is it possible for this person to ever bring the deer into focus using their eyes as they exist?
No.
Just get closer.
No, what does the lens do? We talked about what can
Exactly.
It curves. It curves, but only to a certain point.
Right, but it can curve a bit and pull this focal point in so we can actually bring it into focus. It's going to require accommodation, right? It means normally if you just relax your eyes, the deer is in focus. In this hyperopic eye, you actually have to start using your accommodation to bring it into focus because the eyeball is shorter than normal. But you can bring it into focus, but it means that your eyes are going to be working. Does that make sense? However, what about we look at the flower up close? You know, you are using your accommodation on your eye just to bring the far stuff into focus. By the time you start getting nearer things that are already diverging, your lens has already used up its ability to get more curvy and bend more. So as things get closer and closer, your lens is like, I'm already done. I'm at the limit of what's possible here. You know, so this person can see the deer. That's why they're called farsighted. But when they try to look at things that are closer, they can't anymore because the lens has already used its accommodation power just to look at, you know, bring the far thing into focus. You know, so spend some time trying. If this isn't making sense, make sure it makes sense. You are like on your lab practical. You know, there'll be questions about understanding what is myopia versus hyperopia. You know, so you should understand the basic idea. If I want to have corrective lenses for my hyperopic person here, what kind of corrective lens would I use?
More convex?
Exactly. I would use a convex lens. So now the light rays that are coming in from the thing that are far are going to get pre-bent inward. And then they'll continue on and they'll land right where they're supposed to. And now the eye can function normally. So you have to have a convex lens that has just the right amount of curvature. So the lens causes the lens at rest in your eye will function to bring the image into focus on the retina. You know, if you ever want to know if somebody is farsighted or nearsighted and they don't want to tell you, just pick up their glasses and hold them over a piece of paper. This is a magnifying glass. If their eyeglasses act like a magnifying glass, they're farsighted. This thing makes things look dinky. If you look at things through their glasses and actually the letters are getting smaller on the page, you know that they actually are nearsighted because they have lenses that are concave. These are something different. These are just like magnifying lenses, which are kind of like, I guess, kind of like this. The other thing that can go wrong in terms of getting the image into focus is everything right now we're assuming is radially symmetric, meaning whether you're looking at light rays coming, you know, up and down or side to side, that everything is all symmetrical and that your cornea is completely symmetrical frisbee and your lens is a completely symmetrical frisbee. But that's not often the case. Often you end up having a lens or cornea that is slightly off, is not perfectly symmetrical. And if you have light rays that are kind of coming in up and down, they might converge someplace, whereas if you have light rays coming in side to side, they might converge a little further back or a little further closer. So just because, so this is different than the big picture of bringing something into focus. This is actually the orientation of the line actually can cause things to come into focus further or closer. This gives you what's called astigmatism. This is caused by kind of asymmetries in the curvature of the lens and the cornea. You know, this thing here is what you use to test for astigmatism. You know, I would say do your best at home using this image. And the basic idea is you have to use one eye at a time, close the eye, and then look at it and honestly decide if some of the directions, some of the lines look darker or lighter or more fuzzy or less fuzzy or thinner or thicker. The reality is the image is completely symmetrical, but almost everybody will have asymmetries. Like I look at mine and 12 and 6 axis is just dark and clear, but the 10 and 4 axis is kind of fuzzier and lighter. With my other eye, it's similar, a little different. I think 11 and 5 are my strongest ones. So, you know, in the reality, it's not the picture, it's my eyeballs. So when you get corrective lenses, it's not just doing the near, far, hyperopic, myopic kind of correction. It's also doing this astigmatism correction, trying to get the radial asymmetries dealt with as well. So if in your lab notebook, take a look at this image and just write down what you find in either eye. Okay, you don't have to do it right now, but you know, you will just, and then I see a number of people with glasses, you know, do it with and without your glasses, you know, and the same thing for the near and far, which we'll talk about. How do you measure visual acuity if you're, what your visual acuity is, if you're got emmetropic eye or if you're nearsighted or something? Anybody know? Sarah?
Those are like those letters, right, on the wall, like the E and then like other letters and numbers and stuff.
So here, and normally there is a chart we would have in the room. It's called the Snellen eye chart. And normally you would put it on the wall 20 feet away. And that 20 feet away is important. But Tina found one that you can use, that you can print out using a PDF. So this is something you'd have to do at home. You print this thing out. You put it on the wall. This one is designed to be 6 feet away. You know, so it's smaller than a normal eye chart. And then one eye at a time, do left eye, do right eye. Again, do with, do without corrective lenses. This is, you're gonna need 2 people for it. So again, only do this if there's somebody that can be talking you through it. But the basic idea here is you start reading through, you know, E, LT, FPH, and you keep going until they make a mistake. What was the last line that they could read without making a mistake? And then you note that, and you then have to see what does it say right beside it. Let's say this was the last line they read. They read this next line and they read it wrong. They made a mistake. So this was their best, this was the last line they read well. And then you say, oh, it says 20/40. So what does that mean? What does it mean if your vision is 20/40?
Isn't it like what you can see at 20 feet is what someone else can see at 40 feet? Or is it the other way around?
So that means you're standing 20 feet away. I mean, in this case, it's 6 feet, but pretend you're doing it on a real chart and you're standing 20 feet away and you see with the same visual acuity of a normal emmetropic eye that was standing 40 feet away. So a normal person vision could be twice as far away as you, and they see it as well as you do standing 20 feet away. Right? If this was the last line I could read without making a mistake, I would have 2100, which is not uncommon. A lot of people who wear constructive lenses without their glasses, some of them can't even, let's say all you could see is the E. If all you could see is the E, you would be 2200. That means you're standing 20 feet away and you see as well as someone with normal vision standing 200 feet from the chart. So when you are interpreting your results, make sure you write down what does it mean. Don't just say, oh, I'm 20/30 or I'm 20/20. Tell me, what does that mean to be 20/20 or 20/40 or 20/30? What does it mean? So if I could read all the way to here, what does it mean if I'm 20/20?
You have normal vision.
Right,
you have a very high acuity.
But from using this, what I'm talking about, 20/20, what does it mean in terms of this 20 feet thing?
That you can read that 20 feet away. That line.
But we're always, but remember, what does 20/40 mean?
That you're reading that line at 20 feet as a normal person or person with a normal eyesight reads at 40 feet away.
Exactly. Exactly. So if I'm 20/20, I'm reading
the same as a person with a normal vision reads 20 feet away.
This standing at 20 feet, what a normal person would see at 20 feet. Zing. So I've got normal vision. 20/20 is kind of typical emmetropic eye, because you are looking from 20 feet and you see what a normal eye would see at 20 feet. This chart doesn't actually, it's kind of actually, it's like a drag. It doesn't actually have it here. In the last few moments here, I do have time. I'm going to go, hold on. Images.google. If I look at just a Snellen eye chart, if I look at a normal Snellen eye chart, there's usually another line on it. Oops. Oh, you goofy thing, hold on. I want to go tools. Let's get large. And here's a good one. We're gonna want to view the image. What? Dag nabbit. Okay. Now this is not how, so basically as you get smaller and smaller, you can actually get down to what it says like 2015. So like a lot of times the one down at the very bottom, in fact here, this one will show it. Oh, here you go. You can read this bottom line here. It says you have 2016. What does it mean to have 2016 vision?
It's nearsighted. Well, that he sees or he or she, the person sees like the same as a normal person sees 15 feet away, so closer.
Right, exactly. So you have better than, normal vision, which it can happen. You have better than the average, good eye. So that happens as well, where you can see things with more acuity than 2020. 2020 doesn't mean it's perfect. It means it's normal. You can be 2015. You can be better than 2020. So it's 12:30. We're going to have to finish this lab on, again, we have time because we have all Thursday. We have Tuesday. The review is not just going to take, 3 hours. So what I would do now, between now and Thursday, do the color blindness thing, do the visual acuity, write down your data. We'll finish up the visual system and we'll do these other senses on Thursday. If you have not been kind of catching up on your other labs, make sure you're catching up. We're getting to the lab exam and kind of the final, final grading of the lab notebook stuff. So make sure that you are catching up. And if you're finding you're confused about things, ask. I'm here to help you figure things out. So so that's another thing. Like a lot of people, some of the more successful people have been sending me emails with like, how does this look? And I'm like, oh, that looks good. Or actually you need to think about this a little differently here. So, I'm happy to give you feedback. So I'm going to be here for the next hour for office hours, but in terms of class, we are officially done.